In a previous lesson, we learned what it meant to say A causes B, and this is important because the definition of A causes B will help us design experiments. And in this lesson then, we'll dig into how do we design experiments and also introduce this thinking format that we will use for many cases coming up in the course. We have an if and then result interpretation. Very briefly here, next to the if we write the hypothesis, usually in the form of A causes B. Next to the and we describe the experiment we're going to do to test the hypothesis. Next to the then we're going to write our prediction of what should happen if the hypothesis is true and we do this particular experiment. The result is what are the actual results of the experiment. And then we're going to compare the results to the prediction. And if the results match the prediction, we will conclude, that's our interpretation, that the hypothesis is supported. If the results do not match the prediction, then we have evidence the hypothesis is weakened. So that's the outline of our thinking process, and it helps us really sort of keep track of what we're doing when we do experiments in science. So we're going to, our case study will be, again, the coronavirus. And here I've laid out then all of the different parts of our thinking format. Let's take it one at a time. First up is the hypothesis. So if the SARS-CoV-2 virus causes COVID-19. So again, notice it's a form of A causing B. And for now on, I'm just going to say the virus causing COVID-19 or the disease. So this is our hypothesis, and it's early on, this was an attempted explanation of why people were showing these symptoms. Perhaps there's a viral infection that's causing this disease. Now we're going to introduce some terms here. Whenever we write A causing B, we're going to say that the A is the variable in the experiment that we want to do to test the truth of this hypothesis. So the A is the thing that we want to test to see if it really has the power to cause B. We're going to call that the variable. The B is what we're, in a sense, measuring, what we're looking for to see whether A really is causing this outcome. Okay, now on to the and. Here we write a description of our experiment. So we're going to inject 100 subjects, now these are healthy people, with the virus, and then 100 subjects with the saline solution. Well, the group of people that get the virus, since they're getting the A, the variable, we're going to define that as the experimental condition. So the experimental condition has the variable. In this case, it's the virus. The other group in the experiment, they're getting saline solution, so it looks like they're getting something different, but we want to think of it as they're not getting the variable. So we're going to call that the control condition, does not have the A as defined in our case. That's our variable, the virus. So the experimental condition has the variable, in this case the virus. The control condition lacks the variable, so no virus for these 100 subjects. Notice then there are two conditions in a good experiment here. And this is so that we can see the effect of the variable. We want to compare people who get the virus with people who don't get the virus. We want to compare their outcome. Do they all get sick, or do only some get sick and others not, etc.? So we have to have these two conditions because we want to see the power of the virus and whether it can cause the disease. Now let's go on to the then. This is where we write our prediction. Again, we always assume that the hypothesis is true. Even if we know it's not, for this type of thinking, we always assume it's true for the purpose of making our prediction. So our prediction would be the group getting the virus should develop COVID-19, and the group getting the saline injection should not get COVID-19. Notice we have two predictions here because we had two conditions in the experiment. We had the experimental condition and the control condition. And again, reminder that this prediction is always assuming that the hypothesis is true, and it follows from this specific experiment. Well, now come the results. So then somebody will have to tell us, or we will have to gather our own results. So we get the results. So what happened in the experiment, who got sick and who didn't. And then what we're going to do is we're going to compare the results with the prediction. Now, I've written it down here that the results were the same as what we predicted. But now remember, this prediction we made this prediction assuming the hypothesis was true. So if the results are the same as the prediction, then that's evidence that the hypothesis really is true. So we would say the hypothesis is supported. But again, that we only conclude this if the results match the prediction. If the results are not the same as predicted, then we would say the hypothesis is weakened. And we have a couple of options here. Either we can discard the hypothesis or we need to revise the hypothesis. Here's an example of where the results might not match the prediction. So again, the prediction was the group getting the virus should develop COVID-19. The group getting saline should not get COVID-19. The actual results, let's be careful here, in the group that got the virus, 50 subjects developed COVID-19. So only half the people got the disease, although they were all exposed to the virus. In the control condition, none in the group getting the saline injection got the COVID-19. So notice then, this result is not quite a match for our prediction, because up here we sort of said that everybody in the group should get COVID-19, but only half of the subjects got COVID-19. So it raised the question, what do we do now? Do we discard this hypothesis, or do we need to revise the hypothesis to help us understand why only half of those 100 subjects got COVID-19, even though they all were injected with the virus? Now, as it turns out, with this particular virus, there is an interesting finding that a lot of people can be exposed to the virus but not show symptoms. You may have heard this term, asymptomatic individuals. So they might have levels of the virus in their body, but they're not showing the typical symptoms of disease that others are showing. So scientists are really interested in this phenomenon. Why is it that some people, they can get the virus, but they just don't get sick? We'll use this diagram to go over some possible explanations for why it is that some people get the virus, but then don't get sick. Again, remember, in order for the virus to be successful in reproducing, it has to gain entry into a human cell, and it does it by way of these surface proteins, like kind of keys going into a lock. They have to bind to a protein on the human cell, and that allows the virus to gain entry. Well, for one possible explanation is that the people in the population have slight differences, genetic differences in the proteins that line the surfaces of their cells, and so that may make it easier or harder for the virus to gain entry. But more recently, there has been some evidence that there are differences in people's immune systems. And so that makes a difference when they're finally challenged with the virus, how their body responds. In this picture, we're showing a very important kind of protein that immune cells make. It's called an antibody. It's a protein that is specifically constructed to bind to latch on to a protein on the surface of the virus. And the point of doing that is to block the interaction, so the virus can no longer use its key to get into a cell. So if this protein is covering the surface of the virus because that protein sticks to the viral proteins, then it blocks the ability of the virus to get into our cells. So it may be that there are differences in the human population with how effective our immune systems are in quickly generating effective antibodies to the viral invader. So here we see a list of some possible reasons why people might get the virus, be exposed to the virus, but then not get sick. Interestingly, some recent evidence suggests that previous infection with other kinds of coronaviruses, for example, the common cold is often caused by a coronavirus. The previous infection may be related to how well your body responds to the current coronavirus. And the idea is that by having lots of colds in the past, your body has had lots of opportunities to generate antibodies that may be effective in some partial way against the current coronavirus. Those individuals who have not had many interactions with previous coronaviruses may be more vulnerable. In addition, vitamin D levels may be related to immune function. Those of us who have a vitamin D deficiency may be vulnerable to the virus. In addition, the viral load, that's just how much virus you're exposed to. If somebody just is exposed to a small quantity of the virus, it may be that the immune system has time to sort of win the battle early on. But with a larger dose of the virus, it may be the virus gets an upper hand early on. And then also the genetic differences may make a difference in the effectiveness of the virus getting into our cells. So in light of these factors, we're going to revise our hypothesis. So if the SARS-CoV-2 virus causes COVID-19 in people with immune system vulnerability, so that'll be our catch-all for these differences that make a difference to people getting sick from the virus. And we inject 100 subjects with the virus and 100 with a saline solution. And now here we might stipulate, we might say that, well, we're giving enough virus that for the average person, it would be enough virus to cause illness. Then those getting the virus and have an immunological vulnerability, maybe they have low vitamin D, maybe they haven't been exposed to cold viruses in the past, should develop COVID-19, and the group getting the saline should not. And then when we look at the results, the group that got the virus, 50 subjects with the vulnerability developed the COVID-19, whereas none that got the saline injection got COVID-19. And so then since the results match the prediction, we say hypothesis supported. This case then also illustrates that the world can be complicated. When we have results that do not match a prediction, we're faced with either discarding the hypothesis entirely or revising it. In this case, all we needed to do was revise the hypothesis. There was good evidence that the virus can cause disease, but in order to account for those individuals who get the virus and don't get sick, we had to revise the hypothesis in this manner, that the virus will cause disease in people with immune system vulnerabilities. This illustrates that the world is complex and there are many factors that might be contributing to some particular outcome. But it is the method of science that can uncover these complexities in the world. All right, to wrap up then, an experiment. Here's a couple of key takeaways then. Since a hypothesis is usually a statement like A causes B, we're going to define the A as the variable, and that's the factor of interest. It's what we want to test in our experiment to see if it really does have the power to cause something else, B. The experimental condition has the variable, in this case it was the virus, so we're supplying A to see if B happens. The control condition lacks the variable, we're blocking A in a sense to see if B does not happen. So again, notice that definition of A causing B. Well, the very definition, there were 3 points in that definition. The points 2 and 3 help us basically design these two aspects of our experiment. We're supplying A to see if B happens, and we're blocking A to see if B does not happen. So the definition of A causes B helps us set up a good experiment to test the hypothesis. Then we compare the results of the control and experimental conditions to see if A really did have the effect that we predicted. And again, B should happen in the experimental condition and B should not happen in the control condition. In addition, in this lesson, we introduced our thinking format. It helps us keep track of what we're doing when we're doing scientific experiments. So we have if is the hypothesis, and is the experiment, then is the prediction. We always make that prediction assuming the hypothesis is true and mindful of this particular experiment. Then we get results from the experiment. We compare the results to the prediction. If the results match, we say the hypothesis is supported. If the results do not match, we either discard the hypothesis or we have to revise the hypothesis, as we saw with the coronavirus case.